today. You remember the Petra FROMME also from last week's conference. That was in the opening note lecture. And I think those of you that work now with this field, you start now increasingly and very frequently reading these names in nature papers, science papers, and so on. And I mean, you had part of this story last week. Now today we have Petra with us. Petra is I think she has studied biochemistry. She studied biochemistry, did her master's at the Free University of Berlin, and then for the PhD she moved to the Technical University of Berlin, and then she habilitated. Habilitation is something that is mainly, I think, in Germany, and this gives you the permission to teach in the university. Then I think it was for some period a professorship also in Berlin, assistant or associate professor. Yeah. And after this period in Berlin, Petra moved to Arizona State University, where she became a full professor. And I'm not going to many details of her work, just saying that her main research interests are photosynthesis and infectious diseases, and a big part of this work requires really new methods and new approaches compared to, let's say, conventional approaches of structural biology. And we have seen now that the free-electron lasers are increasingly becoming the tool to approach problems like those Petra is working with. And Petra is going now to tell you some recent results, some of the results and recent results about this exciting new field of femtosecond crystallography. So, Petra.
Thank you very much for the kind invitation, and to the really nice institute. I'm very impressed. You know, we have not remotely such a nice, essentially, institute at the moment. Even if in 3 years we will have actually also a really nice new building. So I want to talk with you about femtosecond crystallography, and you have heard from Raimund, who's my husband and actually works with me on all this project, already the talk at the conference. And so what I will show you today, also we see some similar slides, but I decided to tell you a little bit more about the secrets, how to grow these nanocrystals, how to characterize the nanocrystals, and how to do the femtosecond crystallography experiment. So more matter of the fact, a little bit more into not only shiny pictures, but also a little bit of more how the secrets of how we get to the results. And I hope that will be therefore a nice kind of complementary talk to what Raimund has already given you. So this slide, I think you have already seen several times. And I just want to mention here that a lot of people, when we published papers with 50 authors, said, well, how many of the people do you really know? And I can tell you more than 50 of them, because most times we come together at the free-electron laser and do the experiments together, even driving with the U-Haul truck from Arizona, 1,400 miles, to California, to essentially schlep half of our lab equipment there, which is actually the equipment needed for growing the crystal and characterizing the crystal. And this is what one of the focuses will be of today of my talk. So I want to specifically mention here Henry Chapman, which we have a long collaboration in DESY. And at the end I will tell you also about a really new project which I do with Henry and two accelerator physicists, actually to build another second free-electron laser. So I will be actually for the next 5 years about 50% of my time in Europe and spend significant time actually at DESY. So I will be at ASU and at DESY at the same time. Even if ASU will stay my major tenure, so I'm a guest at DESY. So I specifically mentioned Henry, then John Spence, postdoc, Alexander Hofbrenner, Hugo Weierstadt from ASU, with all our students. And then I want to specifically mention collaboration with Martin Scheretsov and also Martin Kaffel at Trinity College, where we have made the recent discoveries on the G protein coupled receptors. So where does actually all the motivation for this femtosecond crystallography comes from? And it comes from challenging protein complexes. And one of the proteins have a heart beating for us photosystem 1. This is one of the two big protein complexes which in plants they convert light energy into chemical energy by essentially capturing the light by huge antenna system and then making a charge separation. So they're charging the membrane like a battery, which is then used for building up ATP, reduction of protons in form of NADPH, to then build up practically all biomolecules in our biosphere. So it was a big deal actually to try to crystallize this complex in photosystem 1, consisting of 36 proteins and 381 cofactors, is still the largest and most complex membrane protein ever crystallized. There are 499 direct protein structures now in the PDB database compared to 100,000 soluble structures. The membrane proteins lag more and more behind, and therefore new methods are needed to crystallize it. But it took us practically 30 years from the very first small crystals observed by Dr. Galvin and his wife Ingrid in 1988 to get molecular resolution structure at 2.5 angstrom in 2001. And so we really, because this very small crystals we have observed really early on, what took us really so long? And what was actually to get these crystals better ordered and larger. And we need the development of new techniques to determine atomic resolution structures from large macromolecular targets like photosystem 1 with nanocrystals. And just to give you an impression, this thing has a molecular weight of 1 million Dalton, and 30% of the protein is cofactors. So all these things here shown yellow are the chlorophylls, which capture the light and which make essentially plants green. And so we use that actually as the first model system basically for femtosecond crystallography. So before I really want to go into femtosecond crystallography, I want to tell you a little bit more about how we improve the crystals so that we can actually got the normal structure of a synchrotron. We have essentially established a so-called rational design of membrane protein crystallization, where we look as which crystallization conditions are reversible, and then use crystallization as a large purification step. Because when you have these huge complexes, and you have one subunit a little bit nibbled on, there's really no chance that with any chromatographic method you would be able to separate them. Then we determine phase diagram. I have my habilitation in physical chemistry, so I'm a physical chemist. So I hated this trial and error thing, so we really want to know what we are doing. And then we do a lot of biophysical and biochemical characterization of the complex. So for example, light-induced charge separation in the crystals, so we know that we have not crystallized a dead protein, but that the protein makes in the crystals exactly the same reactions that it does in nature, in the leaf, in the membrane, or in the solubilized complex. And this is really important later on when we want to do time-resolved measurements. We have to be sure that the protein is active in the crystals. So then PS1 is also crystallized in a very uncommon way, which actually has been already in the textbook for McPherson for more than 20 years. We crystallize it at low ionic strength. That means you actually decrease the soil concentration until the protein is crystallized. And the effect is the following. When you go to very low soil concentration, you deplete the surface of the protein from the counter ions, and therefore you can get direct crystal contact from a negatively charged group of molecule A with a positively charged group of molecule B. And this is how we crystallize PS1. In principle, all proteins are less soluble at low ionic strength, but because you are going reverse, that means decreasing the concentration of, in this case, soil, you cannot screen this with a crystallization over. And this is the reason why practically nobody tries this nowadays. And this is extremely mild, so this big complex is perfectly stable at low ionic strength, so the precipitant is practically water. So then, when you use purification and crystallization as a last purification step, we just know this is this phase diagram, so we reduce the salt concentration, then we're going to the metastable nucleation and precipitation zone. Then we can make large amounts of medium-sized crystals by more or less going at the unsaturated low magnesium sulfate concentration, and then just concentrating the protein, for example, in an armicron, or even in this small-scale crystallization or concentration devices. That means you approach the nucleation zone by keeping the precipitant concentration constant, and then actually concentrating the proteins. And then you get large amounts of essentially crystals of all different kinds of sizes. So this, for example, is a sample how that stuff from the armicron looks like. Some visible crystals and a lot of this kind of small stuff They even don't know if it is amorphous or it is crystalline. So then, when we want to crystallize it, then we make these big crystals. We actually go from the really small crystals to the medium-sized crystals to the big crystals using actually sophisticated microseeding and macroseeding techniques. So finally, we go close to the metastable zone, add one of these medium-sized seeding crystals, the surface will dissolve, and then we can actually grow these nice big crystals out of it. We have even crystallized 3 times in the space shuttle out of desperation to get better crystals. So we tried practically everything. And this is actually the largest crystal ever grown from PS1. So this is actually a ruler, and this is 1 millimeter. So this crystal is 4 millimeter in length and about 1.5 millimeter in size. And this crystal diffracted to 3.4 angstrom at the time that the best Earth-grown crystal was only 5 angstrom. And we have used it for room temperature data collection. So that's the only crystal we ever got at room temperature full data set by shifting it practically after each image. And this is actually the native data set from the spatial crystal was actually the basis for a better 4 angstrom structure we published in 1999. The problem is you cannot always go to space. And so we then use essentially this phase diagram determination in seeding to also try as good crystals to be grown on Earth as possible. So one of the problems why we need these monster crystals for standard crystallography is the X-ray damage. So when you add a synchrotron and you shine just under cryogenic temperatures, collect just 5 images on this one, you not only lose the diffraction, you actually lose the cofactors. That means you see a white spot in this dark green crystal. These crystals are about 0.5 millimeter to millimeter size. And when you look at the size, you have this large unit cells, 288 by 288 by 167 angstrom. You have about 280 trillion trimer molecules in such a crystal. And you need it because of the X-ray damage problem. This also shows why we get such a big improvement in space, is because this is a horrible crystal packing. We have 78% solvent. This is 1 million molecular weight, and we have only 4 salt bridges involved in the crystal contact. So when you have this kind of horrible packing, it's really low crystal contact. The question is, do we even have nanocrystals? And do they exist? And the first thing which we did actually, so what I wanted to also show you is, when you know take such a big crystal, you see nice diffraction to better than 2.5 angstrom, but you're not able to evaluate the data because of the high long range disorder, even if you do it with seeding or if you do it in space. You see actually all these higher resolution reflections overlap. It means even if we tried really hard for the last 10 years to get an improved structure of PS1, we were not able to evaluate the data because even with all these techniques you can do to improve the quality of these big crystals, we didn't get a better resolution structure. So then we did essentially the thing that we took these small crystals which we get in the Amicron, and in this ultrafiltration step, and filtered it through 500 nanometer filters. And so you get something crappy looking like that. You would say, well, this looks like amorphous precipitate. But we showed this powder diffraction to about 20 angstrom that this is crystalline. And this 500 nanometer crystal is just 17 by 17 by kind of 30 unit cells. And we even used 100 nanometer crystals, which are just containing less than 100 molecules. And we saw still the 3 first order of powder diffraction. So we know that crystals with less than 100 molecules of PS1 exist. But what do we want to do with them? Because powder diffraction is not an option. Because of overlapping of the rings, you will never get essentially a structure that shows you the position of 150,000 atoms with a powder diffraction pattern. And so therefore different X-ray sources are needed. And this is where the free-electron laser comes in. So we had the free-electron laser in Hamburg was actually the first one at low energy, flash, but then the first high energy one, where you can look at atomic resolution on here, atomic resolution structures, was LCL as a linear coordinate light source in Stanford. And it started to operate at 8 angstrom. And the building of this free-electron laser was actually motivated by the idea of Richard Neutze and Janusz Hajdu, that they in 2000 published a paper in Nature where they simulated the explosion of essentially lysozyme in an extremely strong X-ray beam. And they proposed that if you could make something like a super strong X-ray beam, like 10 to the order of 12 stronger than the strong synchrotron, but you would limit it to less than 10 femtoseconds, you might get diffraction of a molecule. before this is stopped. That was actually the idea why they built essentially this free electron lasers. But the problem was that when we worked on the project and tried to convince the referees of our plants that we want to explode these crystals in the free electron laser beam, and there we get diffraction from the nanocrystals, all our 10 grants which we have submitted were rejected. Because the referees said, no way that this experiment will work. The first argument is that they said, look, they didn't believe that you get diffraction of a biomolecule from such a strong X-ray source before this is formed. Second, they said, just assume you will get diffraction. You will never be able to detect that. Because you have 10 to the order of 12 more photons, but you have 10 to the order of 15 less time. And that means your total flux in your pulse is 1,000 times less than when you expose whatever 1 of 10 seconds at a synchrotron. So it means you have 1,000 times less photons, and then you have, instead of trillions of molecules, only hundreds or thousands of molecules in the crystal, in the nanocrystal. So you would never be able to detect anything. The diffraction would be way too weak. And then the last criticism was that they said, even in the likely case you see 3 reflections around the beam stop, you will never be able to evaluate the data, because all the crystals will be in different orientations, different sizes, so it means we will not be able to get such effects from it. So we did the experiment anyway, and it was in 2009 that we were actually just 2 months after the FEL opened, we actually did the experiment. And the interesting thing, we came with 2 samples. One was photosystem 1, the largest, most complex membrane protein ever crystallized, the other was lysozyme. And we had a pretty hot discussion what goes in the beam. Because even our colleagues said, you are completely nuts. Nobody believes that this experiment will work, and you want to put the largest and most complex membrane protein in the beam? Let's start with lysozyme, and then in 5-10 years, when we have established everything, then we can try some crazy samples to do this. But Henry Shipman from Easy is always very adventurous, and he said, there's not really any problem with try this one first, and then it doesn't work, we can always put in lysozyme. So the very first sample, crystalline sample which went into that beam was actually photosystem 1. Unfortunately, it's a little bit dark, but what you hopefully see here is all these nice reflection patterns here. And so all the spots you actually can see now, this is sun coming in. Here, these ones are red, and they're actually overloads. That means our nanocrystals diffracted so strongly that they even separated the detector, which was completely crazy. We have not even expected something like that. So what the experiment works like this, you have here the crystals, and they spray through the beam in a jet in the mother liquid where they have been crystallized in. And what you see here is this glow. And this actually comes from the plasma formed when the X-rays hit the sample. And this we have actually calculated that the temperature you reach when you hit the sample is higher than the incident of the sun. So the referees had a point. They didn't believe that you get diffraction before diffraction. But these really nice diffraction patterns showed that it actually works. And the beam is really so bad that in less than 10 femtoseconds you make hollow atoms that need to rip off first the inner electrons of the atoms, and then you form this plasma. So then what we really saw, and this was really blowing me away, is shape transforms. Zeiler has proposed in 1952 that if you would ever be able to get diffraction from a crystal where you can count the number of unit cells, like 1, 2, 3, 4, 5, 10, in this direction, that you would see fringes between the peaks. And you can count the fringes and you would get the number of unit cells. So you can do this with this crystal and you count to 12 and you see that's 12 unit cells in this direction. So these shape transforms are practically the Fourier transform of the size and the shape of the crystal. And you only have to evaluate one of these shape transforms and you get the size and the shape of the crystal. Like this was a 620 nanometer crystal, and you see even here the hexagonal shape. And we measured more than 3 million diffraction patterns at the very first beam time. And if you would have enough time, you can tell you by the shape transforms how each of them look like. That's not only an academic interesting thing, but John Spencer showed recently that if we would resolve these shape transforms also for the front detector, that means having bigger detectors with more pixels between the peaks, that we would actually be able to determine the phases directly from the diffraction pattern using the shape transforms. So there was 8 angstrom structure which we solved. And then the $100,000 question was, do the diffraction extend to atomic resolution, and do smaller protein crystals have lower or higher disorder? Because you can find, when you have a crystal now with only 100 molecules in it, then many of the molecules are at the surface. So really small crystals may actually have a higher disorder than the big crystals. So we could test that actually just 2 years later in February 2011 when the energy was increased to 1.5 angstrom. That was actually at the time when we published the very first publication on the proof of principle, which is just 3 years ago. So you have to take into account that everything which I tell you now is just happening in the last 3 years. So we have now these small crystals and we wanted to see how they refracted. And this is here actually the beamline here, with us all together working like here, Uwe Weierstahl, with the long injector nozzle, Bob Schumann here from Ilmes group, and here my student, for example, Mark working on it. So that's just part of the team. As I said, only 30 to 40 people come together to do the experiment. And so again, the first thing which diffracted in this higher resolution was PS1. And we got diffraction pattern like that. And I was completely blown away. Working 20 years with this protein and never seen a beautiful diffraction pattern like that. That means these crystals have no disorder. Otherwise, you would not see all these blank regions. Even when you look at the most highest resolution reflections, they're completely separated to the background. So this is just 1 or 2 pixels, 1 or 2 pixels in between, and then comes the next reflection. When there would be any disorder, you would not be able to resolve that. Because the detector is only 1,300 by 1,300 pixels. So these crystals have practically no disorder. And this was really great, but it was also a little bit frustrating, because we realized that for 13 years, we have always solved the really good small crystals, which we used as a large purification step, in order to make the big ugly ones out of them. So we had probably already 13 years these perfect crystals and always throw them, we don't throw them away, we dissolve them to then essentially make the big ones out of them. So it's actually better to make them really small than to go to space. So we have now improved structure at 3 angstrom, and even if the nominal resolution is lower, we are trying to improve it further now. The side chains are extremely well defined for this resolution. So you see all the chlorophylls, side chains, and we even see now new structure elements like lipids or detergent in the crystal structure. And please don't record this because we have not published the PS1 structure yet. So then another really cool thing was in vivo grown crystals. Our colleagues in Lübeck, especially Lars Redeke, discovered that when he expressed the protein, which is the protease involved in pathogenesis of sleeping sickness, so it's actually the protease which degrades your red blood cells when you get infected with it, that this actually, when expressed in insect cells, forms in vivo crystals. That's kind of crazy, isn't it? So you see this crystal here, and this crystal in the cell and even surrounded by a membrane. And so what they did is just breaking the cells, purifying the crystals with the density gradient centrifugation, and then delivering it to the beam. And we got immediately 1.9 angstrom, and it actually shows even the glycochains of the protein really nicely and well dissolved. So this is really cool because this is a natural N-terminal extension that hinders the protease when it is in the pathogen to start its business. That means in the pathogen the protein is inactive. Just when it gets into the red blood cells, this N-terminus is cleaved and the protease starts working and degrading the red blood cells. So this is very nice potential target for essentially treating that pathogen, where about 3 million people suffer from. And we were really surprised actually, because very shortly after we published the paper, we were listed as one of the 9 frontrunners for the breakthrough of the year in 2012 with that paper. We were not the breakthrough because nobody can beat the Higgs boson, but we were one of the 9 frontrunners because of the medical relevance of the protein and because it was the first novel structure elements identified with the free-electron lasers, stereothermal crystallography. So when I summarize now what we have shown you now, I hope I convinced you nanocrystals have superior diffraction quality and then they can be detected by different diffraction methods like powder diffractions. They can be also identified with electron microscopy. And the diffraction quality can be determined by electron diffraction when you have really small crystals. But the ultimate diffraction quality can only be determined by femtosecond crystallography, and for that you will need a free electron laser. And also that nanocrystals have these shape transforms here, and they may allow you phasing in the future. So that's all great. So you may think, okay, I have now all these amorphous specific technology samples. Which of them is nanocrystals? I don't have access to the free electron laser. I cannot really make large amounts of it for powder diffraction. So how could I fast screen for nanocrystals? And this is what I want to bring it up. So you can do high throughput detection of nanocrystals using sonic. And this is actually a new method developed by Galvin and Petra, which uses second-order nonlinear optics of chiral crystals to detect nanocrystals. So how does it work? When you have two infrared femtosecond pulses with infrared light, and when they hit with a distance less than 10 femtoseconds, chiral crystal, that means the crystal of the macromolecule, then you get frequency doubling and get a green photon out of it, and this is what the system detects. And Formulatrix was the first company commercializing this instrument. We were actually lucky because we got supplementary funding from my NIH grant, where we actually got the first of these instruments in academia. So it detects non-centrosemitic chiral crystals. So I mean, some unit cells are actually excluded, but most of them, like P21211, show a very nice signal. And good thing is you get no signals from solubilized or protein aggregates, and because most salts are non-chiral, you get also no signal from salts. So how does it look like? So you have, for example, now here crystallization plates, and you have plenty of amorphous looking precipitate, and then you would be able to, unfortunately, way to light, but you see here and here and here, essentially there are nanocrystals. And probably I can show it better on this picture. Nobody would be able to determine if this is crystalline or not. You would probably think, but this looks like denatured protein. But you see here that it shows up in sonics, that means these are all nanocrystals. And after we discovered that, we started actually collaboration with my membrane protein center for proteins infectious diseases at NIH. This is New York Structural Biology Center of with a group of James Lab. And these are actually the 9 membrane protein centers which were funded by the Protein Structure Initiative PSI Biology from NIH. So we established a collaboration here with James Lab. The good thing is we have the instrument to detect the nanocrystals, and he has high throughput in the insect cell expression. And so we asked ourselves, how frequently may these nanocrystals be in insect cells? Was it just a lucky accident that we saw these nanocrystals, or might this be much more common? So what we did is that James actually selected 11 proteins where he has completely given up of getting any structures from, because there are 5 of them membrane proteins, 4 soluble or membrane attached proteins, but none of them he got any soluble protein or protein out which he can solubilize with detergents. So hopeless cases which of abundant targets. And he recloned them and then expressed them in insect cells and sent us the cells freshly for 24 hours after they were induced. And we immediately next morning measured sonic on them. And this is the result. So this is the blank. And you see now 1, 2, 3, 4, 5 of these proteins of the cells. So this is just taking the insect cells and putting them in the sonic. You see large amounts of nanocrystals and nothing in the blank. So 5 of these 11 samples showed really strong expression of proteins and formation of nanocrystals. And we have actually applied for beam time for looking at these samples. And what we currently also try to do is to see if we can embed the whole cells into a liquid cooling phase injector, which I will show you soon about, which can deliver samples in really viscous medium. Because how cool would be then? We would just express the protein, detect the sonics as their nanocrystals, embed them essentially in a viscous medium, and then directly deliver them to the beam. So no protein purification, no crystallization involves the cells to do it all for you. So that's a little bit pure fantasy because we have not yet been time for this program. So then, essentially we think, that's great, but what does the sonic cost? And unfortunately, after the sonic was such a great success, it doubles the price. So it's close to 500,000 euro, which is sharks from Biodesign no one have for this instrument. And then I want to show you now some secret stuff, which we have not published yet, because there's another way to detect nanocrystals. And this is based on this reversibility of crystallization conditions, which we always use as a kind of last purification step for the proteins. So the experiment was very easy. We paired crystallization plates, we detected nanocrystals with sonic, so to see so if they exist. And then we dilute the reservoir and check the sonic again. And we had 3 samples. One is the protein cap A, fused with GFP, the membrane protein involved in capsule formation of fluorensis, which is actually pathogen-forming rabbit fever. Then photosystem 1, and a GFP soluble protein barrier. And so, so this is how the principle looks like. Here you have the beginning experiment before dilution, and this is how it looks like after dilution. And so you see, for example, here or here, nothing happens. You see it practically the same, or here also. Whereas other drops, the precipitate dissolves. And I can show this here. So that's before dilution, after dilution, before and after. And so the hypothesis was that is there a higher chance that this is nanocrystals than this? So we looked at what is the percentage of the precipitate which dissolves of heavy nanocrystals. So here is now an example of PS1, how it looks like after the different kinds. So after 9 days, we start actually the dilution. Then often the crystals actually get a little bit bigger and then they completely dissolve. Whereas this one here just dilutes, but there's no sonic and there's no dilution. The cool thing is now, this is the result, so it's not published yet, is that when you have precipitate which reversibly dissolves, when you dilute the reservoir, we got for membrane proteins a 90% chance of this being crystals, nanocrystals. And for the GFP, for the soluble one, even 100%. But I would take this as a grain of salt because we don't have a large number of samples here. So 90% of the dissolved precipitates are nanocrystals, and we have confirmed that with sonic. And by using this reversibility of crystallization conditions, you can practically detect nanocrystals even without a sonic. That means when you dilute your reservoir and you see that your precipitate completely dissolves, there's over 90% chance that this are nanocrystals. So that's the good news for everybody who has no sonic. So then we also want to have crystals with as most uniform size as possible. And we have a BLS instrument from molecular dimensions. And this one allows you directly to measure BLS in the crystallization block because it has a reflective mode. That means the laser comes from the top and reflects. And that means you can measure in 0.5 microliter samples directly in the crystallization block. It means when we have our protein, most aggregates actually form during the concentration step. You get a perfectly monodispersed protein from the G-filtration column, and as soon as you concentrate it, you can really transform it and try to aggregate. You can hinder that. You can see that this DLS. So this is a really nice sample here from PS1, where you actually have now nanocrystals when you crystallize it, and these nanocrystals have a very uniform size distribution. So we use this DLS instrument not only to look at the protein monodispersed, but also to see what is the size of our nanocrystals. Here's, for example, a sample that they're all about 500 nanometer size. So using now this concentration method, which I showed you, that means going to high precipitate concentration and concentrating the protein, we have used to crystallize on-site at LCLS a lot of different proteins. All the yellow ones are membrane proteins. So it was a clean time where we actually wanted to test if you can apply these methods to a large number of different proteins. Now for all of them we got crystal diffraction, and for 4 of them actually for the very first time. And I want to show you one example of a protein from our membrane protein center. That's actually a fusion protein between the membrane proximal region of HIV, GB41, and cholera-toxin B. So that's actually a potential vaccine against the HIV. So when you vaccinate mice with this protein, they actually become immune against the HIV virus and neutralize it already in the mucosal membrane. So the problem is that the crystals are only ordered in 2 dimensions, but not in the 3rd dimension. So that means the crystals look like flying saucers. So that means from one side they look hexagonal, from the other side they look completely round. And when you go to synchrotron with these crystals, the only thing you get is looking like a high hexagonal powder diffraction pattern. So what we did then, we crystallized with the same precipitate as a protein under more harsh conditions, getting actually these amorphous precipitate looking nanocrystals, which we confirmed with sonics, etc., nanocrystals. So they're used with the same protein precipitants in batch. And we got at LCLS actually really nice diffraction to about 6 angstrom from this crystal. So this is an example where we have never been able to get any useful crystals, big crystals. But when we make them really small, they can be cut very nicely. So then I want to show you another application, which was another mission impossible when we started. Because I told you we are part of these 9 membrane protein centers, and another of these membrane protein centers is is the GPCR Center at Scripps Research Institute led by Ray Stevens and Vadim Sherezzo. And we formed a collaboration with them and with Martin Kafrek at Trinity College in Dublin, which is actually part of my membrane protein centers, which is one of the 11 PIs of IMBB. And so our idea was that we may want to use femtosecond crystallography to solve structures of G-protein coupled receptors, which make up 60% of all the human drug targets currently with serial femtosecond crystallography. And the idea is that this would be the ideal method in principle because these GPCRs form very readily these mini small crystals. But it can take you sometimes years or even never to get even medium-sized, something like 10 to 20 microcrystals out of this, which are suitable for X-ray data collection at microfocus beam lengths. But even then, you have a lot of problems. You have to harvest them, you have to freeze them, you use a crystal damage, and they normally suffer because they're so small, so heavily from X-ray damage. It means often at the synchrotron, even at cryogenic temperatures, 1 or 2 images of the crystals and they're dead, and then you have to search for the next one and so on. You have to merge a lot of data sets. So we said, how good would it be to actually deliver them directly in the LCP to the LCLS beam and collect the data already on the crystals more or less observed in the first click. First time we applied for beam time, there was no way that you would ever get this LCP to fly. Because a disadvantage of this liquid cubic phase is that it's a membrane-like environment and it has a consistency of toothpaste. So by Uwe Weierstahl, with his students Daniel James and Lin Jivong, actually developed a new type of injector which can actually deliver this gluey LCP phase into the LCLS beam. And it can not only deliver it, it has actually even a big advantage compared to our normal liquid jet, that it uses much less sample. Because one of the other bad secrets actually about femtosecond crystallography is that you use a lot of sample and you measure only 1 out of 10,000. Because the fluid sample runs is 10 microliters per minute. And you may have 120 shots. And when you calculate, you hit only 1 out of 10,000. Now with this LCP, it runs not with 10 microliters per minute, it runs with 110 to 100 nanoliters per minute. And that means you can collect a full data set of about 3,000 images in 5 hours just from 50 microliter sample. And this is much more reasonable because most people that I tell about femtosecond crystallography are so excited and I said, well, for full data set you need about 100 milligrams of protein in form of crystals. I said, okay, never mind, forget about it. But with the LCP injector now, we can essentially with reasonable amounts as small as 0.3 milligrams essentially collect full data sets. And we are working currently also on new media, which allows you to insert pre-grown crystals from membrane proteins into liquid media. And I think Raymond showed you already at this talk that you can do this with soluble proteins, and we try to find it now for membrane proteins. So, when we now had essentially this liquid cubic phase injector running, and we collected data on this GPCR, the next surprise was that these membrane protein crystals diffracted these mini ones, diffracted so strongly that the beamline scientists were worried that we would destroy their detector. How crazy is that? So they were afraid that these spots, because they were so sharp, also no disorder, and so focused, that they would essentially overheat the detector. They were essentially making each pixel that it's hit to bad pixels. So they were so mean that they actually attenuated the beam to 3% to protect the detector. And still you see how very well this crystal is diffract. So we got about 2.5 angstrom resolution, and we are sure that if we could measure with higher intensity, so don't have such a wimpy detector, we would very likely have got better than 2 angstrom, very likely even in the 1 angstrom range from these GPCR crystals. So we are really limited by the signal to noise ratio because they attenuated the beam because of the sensitivity of the detector. So we need better detectors in the future. But we get really nice structures, and this is here the serotonin receptor. And we immediately saw during the beam, that was actually the first structure we solved already during the data collection, that the data is superior. So where at synchrotron several side chains were missing, they were well defined at the XFEL data. And so this is from the paper which we just published in December last year in Science, where you actually see new side chains which were missing before, and you see salt which is well defined. But really astonishingly, we see even different backbone conformation, like the helix here is kinked in the synchrotron, data set, we're straight in the FEL data set, or we see different side chain conformations. And we think that's the reason why what could be the case for this is that in the superconductor relation you have these crystals. This may actually lead to unnatural conformations, whereas in the FEL data set we deliver them in the mother nucleus in the native membrane without any freezing artifacts, and I think that's the reason for the difference. Another thing is we solved recently a mystery story. In 1957, one-eyed lambs were born to sheep at a farm in Idaho, and the Department of Agriculture actually had a 11-year investigation where this comes from. It comes actually from this plant, which is corn lily, and it makes a compound which was then called cyclopamine because it leads to this cyclope-like appearance of these lambs. Actually, they die immediately after birth, so they were not viable. So then people have tried, essentially found later, that this is actually due to the binding of cyclopamine to the so-called smoothened GPCR receptor. And the smoothened GPCR receptor is very important for during embryonal development. But later on in the cell it's not expressed except in cancer cells. So the derivatives of cyclopamine are actually now in clinical trials for cancer therapy. So it was very strong interest of getting actually a structure of this smoothing receptor with cyclopamine. Unfortunately, all the attempts, even if the crystals look really nice, they diffracted only to 8 angstrom resolution. So then we optimize the crystals so that they look like that. So that's something you have to learn, you know. These are the bad crystals, these are the good ones. So optimization means making them smaller, not making them bigger. And so we then collected data at room temperature, and we got a full data set to 3.2 angstrom pellets with 4.3 milligrams of the protein. And this is the structure, and this was also a big surprise because all other agonists or antagonists bind here in the membrane region. Cyclopamine binds close to the cytoplasmic surface and actually completely changes the conformation of these loops. And this may essentially be the effect why it has such a dramatic effect on the embryonal development. And we published this just together with the injector development just this year in Nature Communications. So at the very end, I want to really summarize, because most of you have heard the story already from Galvin, our work towards molecular movies of molecules at work. And this is now essentially getting femtosecond crystallography from the static regime into this dynamic regime. Because most proteins are actually flexible and show dynamic changes, like conformational changes in the catalytic site, receptors docking of essentially the effector molecule, or transporters, transporting molecules through the membrane, or electron transport and energy capturing in case of photosynthesis. And photosystem 2 is actually one of these kind of holy grails of bioenergy because it changed the life on Earth as we know it by converting 2.5 billion years ago our atmosphere, which had no oxygen, into an atmosphere which contains oxygen, and all the higher life on Earth depend on them. And you need actually 4 subsequent charge separation events to extract 4 electrons, 4 protons, from water and resolve, emit 1 oxygen molecule. And all the oxygen in the atmosphere comes from them. So the unraveling of the mechanism of this water splitting complex is one of the holy grails of photosynthesis. And so what we did is actually during one of the first screen times, we already checked diffraction from the PS2 nanocrystals, photosystem 2. And this is actually not a fancy picture showing the best diffraction patterns. That's just my little underwater camera I had for my sabbatical in Australia holding on the screen. And so you measure 120 images per second, but we see only 3 out of 20 here. And you see sometimes you hit a crystal, and sometimes you don't hit a crystal. But you see immediately without any fancy detector calibration or background correction that these nanocrystals diffract really strong, as the PS1 nanocrystals I showed you before. So then we wanted to excite the crystals with 2 laser excitation flashes. So that means we want to get dark and double excited state. We said, why do you try double excited state? The reason is that all spectroscopy shows that when you freeze the crystals, you freeze actually the dark and the single excited state, but you cannot progress towards the double excited state. Also spectroscopy shows that distance between manganese is changed. So we thought, we expected something like medium resolution, maybe 4 or 5 angstrom. And so we thought when we would see a difference, we would see it in the double excited state, not in the single excited state, because there was evidence for conformational changes before from spectroscopy. Working with PS2 or with PS1 paradox is a pain in the neck. That's actually my student Scruples, who was the first author of this Nature paper, which we just published. I think it was in paper just 2 weeks ago. So we have to work really with night vision goggles to essentially don't excite the molecules before we put them in the video, which is a pain in the neck. So then how we crystallize PS2? PS2 is actually one of the cases which you cannot easily crystallize by just concentrating the protein because it contains PEG. And you concentrate then the protein and the PEG and the detergent, which is really bad. So we developed for this PEG-containing samples a different method for crystallization, where we have the precipitant on the bottom, the protein on the top, and then at the interface the crystals form. And when they reach a specific size, they sediment into the precipitant, and then you can harvest the nanocrystals at the bottom of your ethanol tube. We have even now modified the method slightly, where we centrifuge during the crystallization step, so we speed up the sedimentation and formation of the crystals. And so with this one, we can get actually in about half an hour to an hour essentially this nanocrystal sample, which you would normally think, that looks like really crappy, compared to this really nice big crystal. That's the only disadvantage of femtosecond crystallography. All the beauty of looking at your crystals is gone, because all what you look at is this kind of highly amorphous precipitate pieces. But the beauty is always in the diffraction pattern. So you are then much more satisfied when you see the diffraction pattern from that compared to this. So this is sonic of PS2, which shows that this is really nanocrystalline. And we also get a very uniform size distribution, in this case of 1 micron of these PS2 crystals. So this is how we did the data collection. So we wanted to have the dark and light state as close as possible. So what we did is here is the drag of the crystals. We expose a relatively big region of it with a 527 nanometer laser, and we run these lasers at 60 Hertz and this at 120 Hertz. And when we do that, then we get always one light image, one dark image, one light image, one dark image. That means in 1 second we measure 60 dark and 60 light images, all identically. That way we can exclude anything of samples being different or things like that. So this is actually now a picture of the light data, structure factors of the light data set. Nothing can be more boring than structure factors, except when you make a video out of it. This is what I want to show you if you've got it for me. So these are all the structure factors determined from PS2. And so you have to now imagine when you measure data with femtosecond crystallography, each of these individual diffraction patterns is an ultrasound slice through essentially the sphere of reflection. And this is the reason why you have to have a large multiplicity of the data. In our case, 300 for the dark data set, light data set, 600 for the light data set to get decent structure factors. Because most times each of the reflections you never hit in the center, you just hit it at the periphery. So you have to have high redundancy of the data sets to do that. And this is something which not all groups take into account. So you see, for example, in the highest resolution cell which we have, in total there are 3,672 reflections. And we measured them for the dark data set 2 million times and for the light data set 1 million times. This is 3,000 reflections. We have really high multiplicity. So do we really need that? In the future, currently we do Monte Carlo integration. That means when we have one reflection, let's say index 775, and we measure it, let's say, 300 times, and we just average what we have measured. We don't take essentially the partiality into account, or essentially the size of the crystal. In principle, one could try to reconstruct the peak profile, and thereby would essentially need probably a factor of 10 less data. But we are working on this, but it's more complicated than you think, because all these shape transforms practically belong to the main reflection. So this makes the thing more complicated. But in principle, we try to find out what is the partiality of each reflection, and then we may need in the future a little bit less redundancy as we have now. Which would mean that instead of 5 hours of data collection, we may need maybe only 20 minutes. So now when we look at the overall structure, it's very similar between light and dark. So that's dark, that's light, double excited. And so when you see here the overall dimensions, they are the same. And this is astonishing because the unit cell for the light data set is actually 5% larger. Not 5%, 5 angstrom larger than the small, that's the unit cell for the dark state. So the overall dimension of the molecules do not change, but we see big changes actually in the center here where the electron transport changes. So that's the electron transport chain. And this is here actually the electron transport chain. Here is P680. So when the excitation energy from the antenna comes, charge separation takes place, and the electron goes across the membrane, first to one kinome, then to the other kinome. And when 2 electrons have arrived here, it takes up 2 protons and leaves the site. And we think this is the reason why we say see a change in the unit cell constant, because after 2 excitations, we have the kinome left. And we think that is the reason why the unit cell constant changed, because the conformation of these loops on top of it changed. And I don't want to go into the details of this. I want to really focus now on the manganese cluster. And this is a model from the 1.9 angstrom structure from Weir, which is the best structure determined so far. Very likely this is not the real dark state. Very likely this is a highly overused state. But at this resolution, we would not see anything about oxidation state of manganese also. So what you see is that the density in the dark state kind of pretty nicely matches here with the loops and the manganese cluster, the dark state's high resolution structure. So how does now the light state look like? And we see actually large conformational changes. So it looks like this connection here is broken and this loops move away. Then you see a new connection here. So this AB loops seem to move closer. And we see actually a connection of the density here now with this loop. Also this manganese may move, and this actually fits to a lot of models which propose that a second water molecule binds here, and then the oxygen-oxygen bond is formed between this oxygen and this water molecule. So we need a higher resolution structure and much more steps in the process. But it already kind of confirms a lot of mutagenesis results which people have done in the past. So what we have now is the very first low resolution step of this molecular movie, which we just published online at the 9th of July. What we want to do is actually get high resolution structure of all the different assays so that we get a molecular movie of what. This might be possible with the free-electron laser at Hamburg, the big one, XFEL, which will open directly in 3 years. And this will have bunches of 3,000 images, and then we'll open 1 second image feed. And each of these bunches, its pulse will be 3 times stronger than LCS. And so when we have this new instrument, then we can give 1 to 3 light flashes, and then there are 3,000 images. 1 to 3 light flashes, 3,000 images. And at this, when I calculated that we would measure about 1.5 days, we would have about 2 million diffraction patterns from each of these 3,000 states. And then we would have a real movie. We would go from just a little bit snapshots to a real movie. So that means there's a lot of prospect for examples of crystallography. You can do examples of data collection at room temperature of these crystals. You can get time resolution for light-induced process already now between femtosecond and milliseconds. And we have already working on development of a rapid mixing injector. That's all really cool, but there's one major challenge, and this is access to FERs. And also that the femtosecond cold storage is too long for single molecule diffraction. But I don't want to go into this detail. The access to the FER is really a bad problem, because there are only 2 free-electron lasers currently at the world. One is in Stanford and one is in Sakha in Japan. And because it's a linear accelerator compared to synchrotron, only 1 experiment can be done per time. There needs only 2 experiments parallel in the world. Not all these experiments are crystallography experiments, high energy physicists, material science, everybody wants to use this FER. So it's a big trouble. Because many people said, this is so cool. But then they never get people care what they want to do with that method. So to solve that problem, maybe in the future, I started a collaboration with actually 4 of my colleagues in Hamburg, Franz Kantner, Ralf Assmann, Henry Chapman, and me. Came up with this really crazy idea of a new revolutionary attosecond XFEL. So this would be an instrument which would outrun any damage because the pulse duration would be femtosecond, sub-femtosecond in the attosecond regime. And it would shrink the FEL from a kilometer to a meter. And it would reduce the cost from $1 billion to 20 million. You think, that's pure fantasy. How could this work? So the revolutionary concept of that is that we would replace the magnets, which at the current case of the picture of FEL, over half of a kilometer accelerate the electron, so that they form these covalent bunches. So that's here a picture of the current Zaza principle of an XFEL. We have magnets which actually form these electron bunches. That we replace this by essentially an electron beam accelerated by inverse quantum scattering. In a transverse laser field, this high-powered optical lasers. That means you actually then replace the magnets by essentially lasers, and they will shrink each accelerator element from about 1.5 meters to 1 centimeter. They will can shrink the effect. Then the second thing, this is actually the even more risky part, is that we would use a completely different type of X-ray source, electron source. Currently, this fancy FEL thing is, the source of it is practically this 50-year-old linear accelerator, which they still have. It is not very efficient. Less than 0.001% of the energy you put in the field emitted as electrons, and the electrons are randomly distributed. This is also the reason why one of the big costs, running costs of the FEL is electricity. Because one of the best kept secrets of the FEL in Stanford is that it uses 20% of the electricity in the Bay Area. That means it includes Silicon Valley, San Jose, San Francisco, when it runs, which is not very sustainable. So the idea is that we would replace this kind of old technology by essentially a completely new concept, which emits already bunched electrons. That means the electrons would be emitted already coherent, driven also by lasers. That means you would also use high-powered lasers to essentially emit the electrons, and they would be emitted already highly coherent. So that's the idea behind it. And then in the access concept, we would combine this new Femtosecond source, which is highly risky, and especially that part has been not established yet, with lots of spectroscopy, like X-ray emission spectroscopy, to get the oxidation states of molecules, and ultra-fast to the optical spectroscopy, to get the electronic state of the molecules. So if this would work, we get an atomic level 3D structure, an electronic structure, and oxidation states. So we think that's pure fantasy. We are in good agreement with all my graduate students actually that said this will never going to work, and you are crazy applying for funding because they will not fund them. So, but we actually got 40 million euro from the European Research Council, just awarded in December, and we started 1st of August this year to actually build that thing. So when it really works, it will shrink the FEL to laboratory scale, about 1 meter size, okay, with the optics maybe 10 meters. Data collection would be possible even to higher energies than as the FEL currently, which is limited to 10 kV, so 12 kV is actually the absorption edge of selenium, so you would be able to go even to higher energies. Peak brightness would be 80 times higher than LCLS, even if the total flux would be 12 times less, because you have only attoseconds instead of femtosecond pulses. Then we would collect data with 1,000 images per second, so 8 times faster than an LCLS, and would overcome X-ray damage even at the single molecule level. This final goal is 8 to 25 attoseconds. This first version will have 100 attoseconds. And then combined with this new fast detector, as I said, not such a limb, there's a curved one, with a large dynamic range, and combined with all the spectroscopy. And this is here the PI of the grant, Franz Gärtner. He came 3 years ago from MIT to DAISY. Then Ralf Assmann, he came 2 years ago from CERN to DAISY, so he was involved in the 6th boson discovery, and myself and Henry Chapman, which I collaborated with for a very long time. And I cannot give you any results because we have just started with it, but if it really works, then it will solve all that access problem to FELs. So that's the team here at ASU, where we specifically mentioned John Spence, Uwe Meierstein, Alexander West works on the mixing devices, and Rainer Hucke, biologist, which works both in the Membrane Protein Center and in the FEL group for single molecule diffraction. Then I want to specifically mention Christopher Kupitz and Shibo Basu, which are first authors of this PS2 Nature papers, where he worked on the crystallization, and Shibo on the data version. Specifically also want to mention my husband, Petra FROMME, who works with me since essentially the beginning of the Femtosecond project. Here's kind of our big team at ASU. And ASU, the desert actually doesn't look like Sahara. It's actually really nice, and we have also some lakes there. Even if it's not beating your view from the institute, no way about that. So we have no funding. After we got actually the results, we submitted one of these papers which was rejected, and then we got a perfect score. So now we have funding from NIH, NSF, DOE for this project. And very recently, just 1st of July, I got actually promised for a new center at ASU. That is the current Biodesign Center, and they want to actually build a second building, where we will hopefully go 3 years into So that essentially will combine all the groups at ASU which involve this Femtosecond crystallography groups. We had also permission to hire 5 more faculty, different areas, accelerator physicists, computation, and spatial biologists for this project. And we got equipment from the university to build a compact X-ray source. That's not FEL yet, but it's like a synchrotron with microsecond focus. And we want then, when we are successful in Hamburg, together with my colleagues in Hamburg, then build microsecond FEL in the basement of the new building at ASU. And for this one, we need a private sponsor. So if you know any rich person which wants to have their FEL at ASU built, let me know. And I want to thank you for your attention.
Okay, thank you, Petra, very much for this exciting talk. I mean, listening to your talk, first of all, my first idea was that my career would have been completely different, because after years of looking, what I was taught was precipitate amorphous project that I gave up. Maybe I would have a completely different career if these tools were available, but I mean, at the end it's too late probably. So are there any questions?
Thank you for your insightful presentation on this exciting field, which will be promising. I would like to ask you if there is a chance to shine the very same nanocrystal a second or a third time while traveling through the...
Yeah.
Can several bunches of electrons shine, or it's meaningless if you don't gain more information?
I think that's a very interesting idea, and actually John Spence has applied for that. So the idea, which you have probably in mind, is to split the original beam so that you have two essentially images from the same crystal with a time delay. The point is we would have to attenuate the beam to not destroy the crystal. But our idea is when you want to have really ultra-fast reactions, let's say happening in 5 or so femtoseconds, maybe when we attenuate the beam, we might be able to do that. But it would not have the sample consumption, because if you make it 1 out of 10,000 or 2 out of 10,000, that really doesn't have much. But I think with a typical phase injector, we have a very decent sample consumption. So we actually can run it with 2 nanoliters, but you have a huge explosion of this jet when you hit it. So we actually just measured this. When you have the full intensity of the beam, we destroy 100 microns of this LCP phase. So we have to completely replenish that. And so that means most times this is 100 nanoliters per minute, where we use this 0.3 milliliters. But on the other hand, if one can attenuate the beam, that means have really strong reflecting crystals like the GPCR ones, we actually attenuate the beam to some extent, and then we have less, the damage is less, and therefore we can essentially run the injector a little bit slower. So I think the way to go is actually the slowly flowing injector, which may not really help for the time of experiments. Because the time of experiments, you want to have time between the flashes, so that you can essentially have your reaction can proceed. So that's a little bit of a shape of that. For the time of experiment, in most of the cases, at least when you have millisecond resolution, you may have to have time in between. But when you want to measure something with a femtosecond, nanosecond regime, that's no problem. You can use a slow running injector and actually expose it with multiple flashes because in the femtosecond or nanosecond time, the crystal doesn't move much. So that would be actually an easy experiment. So the experiment to get femtosecond time resolution from the laser excitation scheme is actually easier than milliseconds, because then you have to take into account how far the crystal has flown. But in principle, this experiment, if it would work, would give you the light and dark state from the same crystal. That would be really cool. But on the other hand, you're not anymore that you have no damage to it, because you would have exposed it already to X-rays in the dark state. So the idea is actually in this direction to do that.
But with attosecond, for example, does it reduce this radiation damage?
The attosecond laser, in principle, when it is really so strong, it would not reduce the damage. But you would be sure that you have even no electronic damage. With an attosecond regime, you're on the total safe side.
Yeah.
I can also say a little bit about the single molecule diffraction, because a lot of people said, nanocrystals is great, what about single molecules? And I don't have to crystallize at all anymore. The major problem with this is that we are currently too long with the exposure. We had been with Rainer being the PI, where we actually use 100 nanometer focus. They have now one set up where you can focus all these super strong X-rays to 100 nanometers, and we had viruses with 100 nanometers. And we had before measured at a different beamline with 10 micron focus. And we got about, also impressive 80 angstroms. And then we thought, now we have all these photons which before were 10 micron, and the virus was only 100 nanometers, focused to 100 nanometers, same size of virus. So I was so optimistic, saying, maybe we get 10 angstrom, maybe even better. Again, 80 angstrom. That was kind of very disappointing. And the only explanation is because we have currently exposure times of 50 femtoseconds. When the molecule really explodes, like Noyce has proposed this in 2000, in 5 femtoseconds. For a crystal, it doesn't matter, because as soon as the crystal is gone, there is no bright reflection anymore, you just slightly increase the background. That means you get reflection for 5 femtoseconds, and the rest is practically just a piece of noise a little bit. But when you have now a single molecule, and you explode it, and you measure 50 femtoseconds, the intact single molecules diffract as well as all the destruction stages, because you have no back enhancement. And therefore you get fuzzy image practically of the exploding virus. So that's the reason why it limits currently. So then we have now attosecond lasers, with 25 or 100 attoseconds, there can be no damage. You would even be faster than this, you would not make holoetics, you would be faster than any damage. And I think very likely if you get diffraction from single particles, one has to go to the attosecond regime. And so eventually our attosecond laser can also be used for single molecule. Maybe for single molecule proteins it might be too weak, but for single molecule particles that might be actually a possibility. So there's a second reason to go to the attosecond regime. That means really autonomic damage even for single.
If you have a passive detector, would you solve this problem to some extent?
Oh, the detector which can run with attoseconds or femtoseconds, you're right. If you would only detect for 5 femtoseconds, but this would be very difficult. You know, when we had the big opening from the center, then actually the president tried to explain what a femtosecond is, because we had also a lot of kind of friends, general public there. And he made a very nice comparison, which I actually never kind of thought about myself. He said that this difference between a femtosecond and a second is the same as between a second and 32 million years. That means when you go to the attosecond regime, there's a difference between attosecond and a second, and is actually then between a second and 32 billion years, and this is 7 times longer than
billion.
Billion. For the attosecond, it's billion years. That means it would be 7 times longer than the universe exists. So that's actually how awfully short such an attosecond will be. And so that means making any detector, which could be limited to 5 femtoseconds, or even don't think about this attosecond thing, might be an impossible task. So in principle, yes, if you would only detect the first 5 femtoseconds, that would be an option, but I don't think that any of the detectors.
What is the current state of the art?
Currently, this detector is a CS PET detector, which was actually developed for single molecule diffraction. And this is also the reason why it's so such a whim. You know, I have recently met the people from Cornell which have developed it and said, they tell me, you are completely crazy, you burn little holes with your crystals into our nice detector, which was originally developed for single molecule diffraction. But because they have not made any progress, really, any breakthrough with single molecule diffraction yet. You know, you see this 100 angstrom proof of principle, but it's not impressive compared with electron microscopy. The detector was really never intended to be used for crystal diffraction. So the new one, which they developed in Hamburg, will have a much bigger dynamic range, will also be faster, not femtosecond, but also be
faster
means that they can measure 3,000 images per second.
Oh, okay, that's what it is.
So that means a faster repetition rate. Because you have to measure them, you have to read them out, and this is a big challenge actually. So there are a lot of challenges on the detector side, so which has to be solved. But your idea is great, but I don't think that anyone can do that.
But I mean, is there any also, I mean, concentric detectors any possibility of developing something that you can also use for phasing from the shape function and so on?
Yes, so we actually have applied for beam time and we got a test beam time. So my colleague Alexandra Ross from ASU, the one working on the microcrystals devices, she actually found, we have a paper that we can sort crystals by size using dielectric-photonics. So he takes a mixture of the, let's say, between 100 nanometer and 500 nanometer crystals, and then all the small ones reflect in the side channels. And so you can make samples which contain only crystals which are only 100 nanometers, plus minus 20 or so. So only the really, really small ones. And we got kind of 5 hours of green time in November, which is not much. But where we will try to see how this looks like, because we want to have a nice data set collected, the detector as far away as possible so that we can nicely resolve the shape transforms, that we have a good data set with good shape transforms. And then we will try to see if we can face an experimental with the shape transform. So we're planning to do that experiment. And we want to use specifically sorted crystals out there that we expect a very uniform size. And a very uniform size, and also when the crystal only 100 nanometers and the beam is 1 micron, we will always have in very good chance to have in each shot just one crystal, so not just an edge of a crystal or something like that. The crystals are the same size as the beam. So that's the experiment we plan in November, and we hope to get a really good data set with lots of shape transforms where we can test that experimentally. Theoretically, the paper from John Spence looks very convincing that you can do this phasing, but the devil is always in the details and in the practical experiment. So we want to get a test data set to see if we can do the phasing. Because this would be so cool if you could do that.
Because that would solve the second problem, crystallography. Crystallization is one, phasing is the other one. Then
yeah, and at the moment it's actually a very serious problem with phasing.
Yeah.
Because some other kind of naysayers said, no, we solve all these structures, but they also have this molecular placement. What is this experimental phasing at an FEL? And shape transforms is one possibility, currently limited by the detectors, and essentially making this kind of samples with all these really mini crystals. Second is the problem that in order to get good phasing data, you have to measure the structure factors very accurately. So having essentially all these partials is not helping you very much. The second major problem is that with the current FEL and the Sartre principle, the energy of each pulse is slightly different from the next one. That means we have to evaluate each image with the wavelengths by which it is exposed. So if you would go to an absorption edge, you would always wiggle around the absorption edge. Second problem is that the peak is actually not a real peak. So you would like to have a peak like this, but in reality, the Sartre, it looks more like this. And between shots, they have now actually installed a spectrometer where you can actually get the spectrum from each shot. And after I saw this, I was pretty much horrified because not only this wavelength changes, but also the size and the location of the site changes from shot to shot. So that's also something which might be difficult for phasing. So Image Lichting published a paper in Nature, I think in December last year or so, November, where she showed this gadolinium lysozyme that in principle you should be able to do the phasing. But you know, gadolinium is so heavy and lysozyme is so small that's not a really good proof of principle. So I think one of the possibilities is to use a seeded beam, but then the intensity is only this, so 10% of the max intensity. And only 5 to 10% of the shots are seeded, so you throw away 95% of your data. Again, the other second laser, if it works, it will have only one wavelength. So it will more or less look like that. So this could also solve that problem. And we can go to, currently we have actually applied for Biodesign to try to use selenium-methionine phasing. And they told us that it makes no sense because when we want to go to 12.5 kV, we have only one 10 to the order of 10 to the minus 4 of the original intensity left. So the FEL is so weak at 12.5 kV that we have no intensity. So that means that's also the reason why we have proposed the upper second laser to go to 12.5 kV, that in principle one would be then, with this system also be able to do conventional phasing like a simple one. This selenium measuring essentially light, measuring SE derivative and native data. And also with much better precision because we don't have this wiggling of the beam. So experimental phasing is at the moment still a problem. I have to admit that.
If we have precipitation sample, how we can distinguish the nanocrystals by the relation with the reservoir, or there is another manner?
As I said, the best way is to do sonic. This second order, so this magic instrument which makes the green photons out of 2 infrared ones. That's very easy because you just put the 96 wave plate in the sonic and you see more or less by R, you see whatever A2 is nanocrystals and F6 is nanocrystals and so on. So that's a very nice tool, but it's very expensive. You pay 200, I think, no, 350 euro, no, dollar, and now they want to have 500, so it's really crazy. Very easy method is the dilution thing. It's the dilution of the reservoir. And so that's pretty easy. So when you have the 96 well plate, we remove the cover slip, and we have a Rigaco crystallization robot, so the scales, which has this 96 syringe heads. So it takes the cover off, and then the system has a new program, which Rigaco will make actually available to everybody else, which dilutes the reservoir. So it takes, let's say, 25 microliter out of the reservoir and puts 25 microliter water in. And it takes less than 2 minutes, and then you close it. But when you don't have the system, then you can use it with 12-channel pipettes. So you will take your plate off, you just, maybe with 2 people. One person has a water pipette, one person has the other one. Out, in, out, in, out, in. I think when you do this by hand, 2 people, you can also do this in 2 minutes. You know, it's a 12-channel pipette, and then you close it. And I can tell you, it works. We did the experiment with 200 nanoliter drops, 100 nanoliter proteins, 100 nanoliter reservoirs, and we used a deep well plate. So when you have the plates, then you have the ones with the shallow reservoirs, or you have one with a more deeper reservoir. Most people like this because it's easier to harvest the crystals, but when you have a drop here, this evaporates much faster. And in Arizona, we have practically 0% humidity. So that means we use, this is the worst case scenario. So we always use these plates, so that, and then we can, in 2 minutes, we have a 200, also 100 plus 100 drop does not dry out. So you have 2 minutes to do that. It's a high humidity here, which is bad for you, but it's very beneficial for your crystallization experiment. So I'm pretty sure when you practice that, first with colored solutions and have your colleague already the water ready, you know, you can do this also in 2 minutes. You take the plate off, you dilute all the reservoirs, taking just 25 off water in, 25 off water in. I think you can do that in 2 minutes. Maybe you have to do this high humidity in 4 minutes time to do that. Then you close it, and then you put it back in your imager, and then you will see which of this amorphous precipitate is soft. And this is 90% chance that it's nanocrystals. And what I didn't say is there's another advantage of identifying the nanocrystals. We also make standard crystallography because the beam time is so limited. We cannot just sit half a year around until we have beam time at LCLS again. So we do also a lot of standard crystallography. And for this one, we screen all plates to see if we have nanocrystals. Or in your case, you would look if you have any amorphous precipitate which dissolves. And then you know, aha, this will be nanocrystals. And then you use this for fine screening. That means when we have these membrane proteins, we have 12 different screens which we normally use. And I don't recall that we have ever had any crystals in any of these first initial screens. It was membrane proteins worst case scenario. You must have 12 screens, and all you get is amorphous precipitate. That's something we have faced with PS1, with PS2, and so on. And so we always look for this reversibility thing. Like before, we always did this painstaking use this next thing. I don't know if you've ever used the screw caps and things like that. Now with the 96 well plate, it's much faster. And so every plate which just looks, forget about it, only amorphous, you can use for that reversibility test. And we do it 3 times. So we take a plate off, dilute all the reservoirs, close it. Let it stand 3 days. Then we image it and then we repeat it. So we repeat it 3 times. And then we kind of compare always the same images. And then you see immediately, aha, H5 dissolves and G10 dissolves. And then you know, aha, this might be nanocrystals. And then you can do 2 things. You can fine screen surrounding these conditions. And you can also use these small, potentially nanocrystals for seeding. That means you can then make a mixture of these little seeds and add them to your crystallization drops as seeds. And that might be also a good possibility then to go from the nanocrystals to bigger crystals that you can measure at more simple form. That means this nanocrystal identification is both important for optimizing to make bigger crystals for standard crystallography and for Femtosecond. So you should really try that. We have not published this method yet. I just got the manuscript from my postdoc, but we hopefully submit it in this kind of next 2 or 3 weeks, and then you will see all the details. But it's actually as easy as I tell you. You just dilute the reservoirs and see if the crystal is off. And then you have a really good chance of finding nanocrystals.
Do you recommend using powder diffraction then to evaluate
Yes.
the most promising stuff?
Yeah, this is another thing which I have not told you. I told you that we use these crystals for PS1 as a last purification step. What we actually do is, when we have identified a condition where we know nanocrystals form and we can dissolve them and we can precipitate and we can dissolve them, what we try to do is to bring the complete protein in batch then in form of these crystals. That means we then in batch or with the Amicon, more or less as soon as the protein is freshly isolated, we crystallize it. And then we often do this 2 or 3 times, and then you get rid of a lot of contaminants. Another secret which we have also not put in our papers is, when you do that, you can relatively start with a relatively dirty protein. Like photosystem 2 is very sensitive. You know, on the plant in the bright sunlight, this is one which makes oxygen, lives only half an hour. That means, and then it has to be replaced. Practically 50% of all the energy of the plant is used for replacing PS2 every half an hour. So that's a nightmare, because in the native membrane, up to 50% of the protein is under reconstruction. So the dimer falls into monomers, extrinsic subunits disassemble, membrane proteins disassemble. Then the protease degrades E1. This is the protein binding the manganese cluster and most of the cofactors of the electron transport chain. Then the ribosomes bound to this partially disassembled PS2 resynthesize the protein. It's like a heart transplantation every half an hour. Then the thing is assembled, lives another half an hour, and is again dead. So it means you have a lot of heterogeneity of the PS2 in your prep, even after purification. In addition, you can have up to 30% ATP synthase as a contamination. The problem is when you now try to doing several column purification steps to get rid of this partially damaged PS2 or ATP synthase, they all co-purify together. So we tried a lot of time to get rid of them and obviously end up with a dead PS2. So we solved that problem by directly after we have isolated it, in this case with PEG, precipitating the protein 3 hours, then dissolving it again, precipitating it again. And in each of these precipitation steps, because the protein gets purer and purer, you need less and less precipitant, you get 90% precipitated. And then after the 3rd purification step, you have this highly oxygen-avoiding activity, active PS2, completely get rid of the contaminants and of partially assembled PS2. That means we use then crystallization as the last purification step, also for the photosystem 2. So it's also what you can do, because we never freeze our proteins. Neither PS2 or PS1, if you freeze it, you cannot get any crystals anymore, because it induces aggregation and all these bad things. So, and we store the protein in the form of crystals. And PS2 is stable for 3 weeks in the dark, even at room temperature in form of crystals, whereas in the native membrane it survives only half an hour. So we have dramatically destabilized. And when you shine light on the crystals, you see actually little oxygen bubbles coming out, so you know it's fully active. That's nice. So therefore, my recommendation: try this reversibility and try to actually eventually use it even as a purification step. So when you see any problems with heterogeneity, you may get rid of it. And you get actually the much better protein. And you can eventually then what you get at the end directly use for Femtosecond crystallography. Or you can dissolve it and make big crystals out of it.
Okay, any other questions? Okay, thank you very much.